Hello everyone, welcome to the channel Biomites. So today we will have the another session on immunology. So we have already started with the antibody series. So here is the third part. So where we can discuss now we will have the continued series on the different types of antibodies. So before this lectures we have already done the two parts. In the first part we have the introductory part of the antibodies. In the second part we have the function of the antibodies, and in the third part we are starting with the different types of antibodies. So if you haven't watched the first two parts, then I uh, tell you to first watch this two part and then we'll continue with this third part. Okay. Okay, so here there are mainly five types of antibodies which are present in our body. So IgG, IgA, IgE, IgM, and IgD. Okay, so why it is known as the IgG? Okay, so as we have seen in the part 1 introductory part, the gamma portion of the serum contains the immunoglobulins. Okay, so Ig stands for immunoglobulins. Okay, so here IgG, so we have these different types of the IgG. Okay, we will study this one by one, but in the different series. So today we will just focus on one antibodies known as the immunoglobulin G, IgG antibody. Okay, so why this different G MD is mentioned is because there are two chains which are present in the antibodies which we have already discussed. So we have the variable region and the constant region. Okay, so here you can see, okay, here you can see there is a, see here, there is a C gamma. So gamma, that's why it's a G. For A, it's alpha. For M, it's the mu. Okay, so for E, it's the epsilon. For D, it's a delta. So for G, it's a gamma. So this is the type of the light chain that IgG is required. Okay, constant region of the light chain, which is the variable in every antibodies. We also have the lectures on the detailed lectures on that. Okay, so stay tuned for that. Okay, so here we have a 2 gamma heavy chains, see here you can say, and 2 kappa or 2 delta light chains. Okay, so IgG in the human we have a 4 different subclasses of IgG. IgG 1, 2, 3 and 4. So IgG 1 which is in the highest concentration in the serum, whereas IgG 4 is in the lowest concentration. That's why the numbering is 1, 2, 3, 4. Okay, so the amino acid sequence that distinguish these 4 IgG subclasses are different germline CH genes whose DNA sequence are 90 to 95% homologous. Okay, so they are encoded by different germline CH genes. Okay, so this is the basic structure of the immunoglobulin G. So 2 gamma heavy chains, okay, 2 gamma heavy chains and the 2 delta or kappa light chain. Okay, so the constant region of this heavy chain is the variable region. So here you can see the different subclasses of the IgG, IgG1, G2, G3, G4. Okay, so the difference is only you can see in the diagram itself. There is a disulfide bond between this heavy chains, 2 heavy chains. So that disulfide bond varies between these two, this 4. Okay, IgG1 has only 2 disulfide bond. IgG2 has 4 disulfide bond. IgG3 has 11 interchain disulfide bonds, whereas IgG4 also has the 4. So it's the same almost. Okay, so the size of the hinge region and the number and the position of interchain disulfide bonds between the heavy chains, so they are only differ between the structural characteristics. Okay, here is the quick function of the IgG. IgG mainly IgG present in the most abundant form in the serum. Okay, so there is a amino acid difference between these subclasses of IgG that affect the biological activity of the molecule. We have already seen the basic function of the antibodies. So here you can see IgG is the only antibody that can cross the placenta. Okay, and play important role in the protecting the developing fetus. But only IgG 1, 3 and 4, these three subclasses only crosses the placenta, except IgG 2. Next, IgG3 is the most effective complement activator. So what is complement that we have seen in the part 2. Okay, so IgG3 is, they can activate complement. IgG1 after IgG3, IgG2 is less efficient, but IgG4 is not able to activate complement at all. Okay, so during the phagocytosis process, IgG1 and G3 has very high affinity. Okay, that does, it can mediate the opsonization. These terms, phagocytosis, opsonization, we have already covered in the part 2. IgG4 has an intermediate affinity and IgG2 has extremely low affinity. Okay, so these are the basic overview of your IgG. For the rest of the antibodies, we will have the another session. Okay, so stay tuned for that. Thank you, and for next lecture, please subscribe and share it with your friends. Thank you.